so they aren't quite so exhausting, I guess would be the word. Okay, so folks, with metabolism, this is a really big unit. For me, studying biology, metabolism was the hardest unit for me to kind of digest, get at kind of a gut level. So if it is proving a difficult subject for you, just recognize you're just normal, that's all. And eventually it will come. It takes a lot of repetition and looking at it from different perspectives, but you will get it. So a subtitle for this unit is energy flow on the planet of the microbes. So a lot of what we'll be discussing once we get through this introductory PowerPoint A will be energy flow and ATP synthesis. So to give us the big kind of roadmap for energy flow, we could think of the sun, solar energy, light energy is a huge source of energy for life on Earth, and we want to remember that we are indebted to the photoautotrophs, such as organisms that can carry out oxygenic photosynthesis, because what the photoautotrophs can do is take inorganic precursors, and in the case of oxygenic photosynthesis, it'll be carbon dioxide. This will be the source of carbon for our little microbes, and then a source of electrons will be water. And in the presence of chlorophyll A and light energy, these little photoautotrophs, they can use light energy to energize electrons from water, and through an incredible series of steps, eventually get those high energy electrons donated to what used to be CO2 to make organic molecules rich in chemical energy, such as glucose. Now, glucose is chock full of energized electrons, energized by the sun, and then the other end product is molecular oxygen. We'll talk a little bit about oxygenic photosynthesis right in the beginning and then a little bit towards the end of Unit 4, but most of our time is going to be used talking about chemoheterotrophs and how they make ATP. And the reason we do this is the pathogenic microbes that invade other organisms, they're chemoheterotrophs, and the reason they're invading other organisms is chemoheterotrophs require a preformed source of organic molecules as a source of energy and carbon, and other organisms, including humans, are wonderful sources of preformed organic molecules. So they're just using us as fast food, and it's nothing evil or sinister. And since we're going to be talking later a lot about aerobic respiration, I always think it's kind of interesting how I wrote this summary reaction for aerobic respiration in reverse. And I did this because it almost looks like the opposite of oxygenic photosynthesis. It truly is not. It's an entirely different process. But still, the similarities I thought were cool. So in aerobic respiration, the chemoheterotrophs are slowly going to rip off the high energy electrons from, for example, glucose. When those electrons are transferred, they're going to release some energy, and it's that energy that the cell is after. The cell's going to use that energy, this energy release, to help drive ATP synthesis. That's one of the big themes in our metabolism unit. But this must be done in the presence of molecular oxygen. That's why it's called aerobic respiration. And it's because this molecular oxygen is going to act as what we call the terminal or final electron acceptor at the end of that electron transport chain. Okay, so in aerobic respiration, if these are our reactants, then our end products are energy, and about a third of the energy will be used to drive ATP synthesis. And then we'll have water. This is when molecular oxygen acts as the terminal electron acceptor, it gets reduced to water. And then once we've oxidized our glucose, we're going to end up with carbon dioxide. Again, just a little bit on photoautotrophs and oxygenic photosynthesis. Okay, so photo means they use light as an energy source. Autotroph means literally self-feeders. They're going to use inorganic carbon dioxide from the air as a source of carbon to build their own organic molecules. So if you're going to carry out oxygenic photosynthesis, you require the beautiful green photosynthetic pigment chlorophyll A. And chlorophyll A is made by cyanobacteria, like nostoc that we looked at in our lab, algae, and green plants. Remember, cyanobacteria are bacteria, so they don't have chloroplasts, but they do have chlorophyll A. Algae and green plants are eukaryotes, so algae and green plants have chloroplasts. And we want to remember the chloroplasts are evolved from primitive endosymbiotic cyanobacteria. So again, most of our time will be spent talking about chemoheterotrophs, and eventually we're building up to aerobic respiration. So again, chemoheterotrophs require preformed organic molecules as a source of energy and carbon. And we'll be exploring 3 different ways that chemoheterotrophs can use the energy, the chemical energy from those organic molecules to make ATP. And the processes we'll discuss, and this will be in the second PowerPoint, be fermentation, anaerobic respiration, which is possibly going to be a new process for many of you because it's only carried out by prokaryotes. And then finally, the process, those of you that have had A and P are really familiar with aerobic respiration. Chemoheterotrophs include all members of kingdom Animalia, all members of kingdom Fungi, some of the protists, and again, you guys, this should be little p without the A on the end, just protists. And the protists, which are chemoheterotrophs, are the animal-like protists, the protozoa, and the fungal-like protists, the slime molds and water molds. And in addition, many members of domain bacteria are chemoheterotrophs. And remember, folks, all pathogenic bacteria, and I should have said all bacteria that invade other organisms and cause harm, are all chemoheterotrophs. And again, the reason they do this, they need preformed organic molecules. You and those other organisms are wonderful preformed organic molecules, so we're just fast food for them, that's all. And then some members of domain Archaea are also chemoheterotrophs. And again, folks, most of our lecture time will be spent discussing chemoheterotrophs. The next slide, you guys, is really disturbing, but I wanted you to see some of the consequences of when microbial pathogens invade us and use this as a source of preformed, use this as a source of preformed organic molecules. And this is really disturbing, but again, I want you to know that metabolism is an important part of pathogenesis, the ability to cause disease. So this is a so-called necrotizing fasciitis, a flesh-eating disease. It is caused often by specific strains of Streptococcus pyogenes, also known as group A streptococci. And then some strains of Staphylococcus aureus can also cause this, but it's most commonly associated with the group A Streptococcus pyogenes. And again, folks, here's an example of a bacterium using us as a source of fast food. So just the rest of this PowerPoint will be truly an introduction to metabolism or developing our vocabulary and concepts. So first of all, what is metabolism? It's all the chemical reactions which occur in a cell. And there's 2 basic types. Catabolism, when we take large molecules and break them down into smaller ones. And then the opposite is anabolism, or biosynthesis, when we take smaller units, smaller subunits, and we link them together to make larger units. Now, usually catabolism releases energy, and usually anabolism requires energy. So we can link these 2 processes in a cell. And this is a cartoon showing the linking. So with catabolism, we're breaking down larger molecules, releasing energy. Some of that energy can be captured to make ATP, right? And then if we look at anabolism, it requires an input of energy. We're taking smaller molecules and making larger ones. And very often the source of energy is going to be ATP. So why is ATP so important? It is often called the energy currency of the cell. A good analogy is it's like the dollar bills of the cell, right? So you can use dollar bills to pay for everything, right? So if we look at the structure of ATP, adenosine triphosphate, not only is it the energy currency of the cell, it's also a building block for RNA, so it serves 2 functions. So here is our 5-carbon sugar, our ribose, our adenine, and here are the triphosphates, the 3 phosphates. The tilde sign is used to indicate a covalent bond which upon hydrolysis will release a fair amount of energy. I was always brainwashed into calling these high energy bonds, so when they're broken, lots of energy is given off. But Dr. Zenner told me, they aren't high energy bonds. You want, relatively small amounts of energy to be released when these bonds are broken. Because remember, they're dollar bills. They aren't $100 or $1,000 bills. So the cells then can use ATP as a source of energy for cellular work. And examples of cellular work are anabolism, also known as biosynthesis, active transport, and motility. These are all examples of cellular work that the cell can use ATP to drive. And these are just the reactions. Again, the cell is constantly using hydrolyzing ATP to ADP, and this is the inorganic phosphate that release, and the cell's after that energy. But if the cell's constantly spending ATP, the cell has to constantly be making ATP, and it does that by a process called phosphorylation. Phosphorylation is when we add a phosphate group to a molecule. So this is PowerPoint B. We're going to spend most of our time talking about how ATP is made. We have ADP, inorganic phosphate, and the cell has to have an input of energy to drive the ATP synthesis. And again, this is a cycle that occurs over and over and over in the cell. We make ATP, we spend ATP, we make ATP, we spend ATP. So the cell is constantly making ATP. In the process of ATP synthesis, we have energy transfers, and in chemoheterotrophs, the energy will be provided by our preformed organic molecules. And this, for me, was like a tough concept. I'm really visual. I want to be able to cartoon everything. So energy transfers, for me, is like, how do I cartoon energy? So Dr. Laurel Heffernan over at Sac State was my teaching mentor when I first started teaching. And I thought she had a really effective way of describing these energy transfers, and I want to share it with you. Now, from like if we were high-level biochemists or biophysicists, this model might break down. So it's probably a little bit more sophisticated than how I'm going to describe it. But I just know for me, when I was trying to master or get a good sense of metabolism, this model Dr. Heffernan used really helped. So you guys, for us, we're going to say when we're talking about energy transfers within the cell, we're talking about electron transfers. And so we're going to describe electrons as little packets of energy. So as the cell moves electrons from one molecule to another, we can describe that as an energy transfer. Now, electrons can be transferred as just like naked electrons, just an electron, or they can be transferred as part of a hydrogen atom. Because remember, a hydrogen atom is a hydrogen ion, a proton, and an electron. So in our reactions, we can watch the transfer of electrons or hydrogen atoms to observe the transfer of energy. And again, you guys, the reason this is important is electron transfers are crucial in ATP synthesis. So from chemistry, these two types of electron transfers. For us, in our introductory biology course, we're going to say oxidation is the loss of electrons or the loss of hydrogen atoms. Reduction is the gain of electrons or the gain of hydrogen atoms. And this was really cool, you guys. When I first started teaching here at Sac City, the micro students, they said, oh yeah, just remember Leo Ger. And I was like, what's Leo Ger? So it's a little mnemonic to help you remember that oxidation is when we lose electrons. So Leo, like a lion, and reduction is gaining electrons. So G-E-R, Ger. And I like to think of this, the lion says Ger, Leo says Ger. And you can come up with any other mnemonic that helps you remember oxidation and reduction. Now, in cells, it's important that oxidation and reduction reactions are coupled. And the reason is it's really dangerous for a cell to remove a naked electron from a molecule and just throw it into the cytoplasm, because those electrons cause all kinds of damage. So within the cell, whenever there's an oxidation reaction, there's always a reduction reaction. That electron has to be transferred to another molecule. And these coupled reactions are called redox reactions. And this is just a generic redox reaction. Originally, the artist just showed transfer of an electron, but I added my little hydrogen atom here so we can remember that electrons can be transferred as hydrogen atoms. So here are our reactants, A, H, and B. And this arrow represents the electron rearrangement. So we'll see that AH is going to transfer an electron, a hydrogen atom, to B, right? So as a result, we would say that AH is oxidized. This is our oxidized molecule here is an end product. And B is going to be reduced. It's going to accept the electron. So AH lost the electron, lost the hydrogen atom. B was reduced. It received the electron, it received the hydrogen atom. Now this is kind of boring, so let's go to the redox reaction that for sure, folks, will be one of the short answer questions on our lecture exam too. So this is a summary reaction for the complete oxidation of glucose by aerobic respiration. So over here we have our reactants. Here's our glucose, here's our oxygen. This arrow represents the many steps that happen in aerobic respiration when the high energy electrons slowly are ripped off of glucose. Okay, so we want to ask ourselves which of the reactants will be oxidized, and that's going to be glucose. So glucose is going to be oxidized, it's going to lose its high energy electrons, and we'll end up with carbon dioxide. The reactant that will be reduced is molecular oxygen. It's going to accept those electrons after they've lost most of their energy. Now they're tired out, low energy electrons. So the oxygen is going to be reduced to water. Now remember, folks, this is what people forget. Another end product is the release of energy. That's why the cell is carrying this process out. And again, about 1/3 of the energy will be used to drive ATP synthesis, and 2/3 of the energy will be released as heat. Okay, so I think we've answered that. Right, okay, so I think we're good there. Now, electron transfers in cells often involve what we call electron or hydrogen atom carriers. And the two I'd like you to know for our lecture exam 2 are the coenzymes. A coenzyme is an organic enzyme helper called NAD and FAD. So NAD and FAD, again, they're called coenzymes. And we always want to remember, you guys, that many of these coenzymes, and NAD and FAD are examples, they either are vitamins or they contain the components of vitamins. Vitamins are organic molecules we can't synthesize for ourselves, so we have to swallow them, we have to ingest them. So having a well-balanced diet with a proper balance of vitamins is going to be really important for our cells for ATP synthesis. And you don't have to memorize this table, folks, but just showing you that for FAD we need the vitamin B2 riboflavin, and for NAD we need niacin, nicotinic acid. This is kind of awkward when we look at the oxidized NAD and then how it's reduced. So notice, you guys, that the oxidized NAD, it has a positive charge. So oxidized NAD combines with 2 hydrogen atoms. It accepts one entire hydrogen atom, and then it steals the electron from the second hydrogen atom. So our end products then are the reduced NADH and a hydrogen ion. What I'd like you to remember for the exam is that the reduced NADH is carrying 2 high energy electrons, which later can be very useful in ATP synthesis. The reaction for FAD is much more straightforward. FAD can accept 2 hydrogen atoms, so here's oxidized FAD combines with 2 hydrogen atoms. Here's our reduced FADH2. And FADH2 also carries 2 high energy electrons. And just for the back burner, you guys, just for us to remember later, the high energy electrons carried by NADH are higher energy than the electrons carried by FADH2. And we'll see later that that will influence how much ATP can be produced. And again, these electron carriers are going to play a role in ATP synthesis. So folks, I think what I'll do here is stop, and then we'll do a follow-up audio talking about enzymes, and that's quite a long section, especially when we get into enzyme inhibitors. Make sure you have your enzyme inhibitor worksheet out, because the enzyme inhibitors get really complicated when we start talking about beta-lactams and beta-lactamases. Okay, folks, so we'll close here, and we'll do another PowerPoint later.